Rebecca, since we have the PBS recording and my formal presentation in there, why don't we start here with a little bit of informal introduction. And I usually ask people 5 questions. I'm going to tell you what they are, and then I'm going to ask you those 5 questions. And the 5 questions are, where were you born? Where'd you go to high school? Where'd you go for your undergrad, and what'd you study? Where'd you go for your advanced degrees, and what'd you study? And then when did you come to UW-Madison? So are you there, Rebecca?
Yes.
Good. Hello. Good to see you.
Hi, everyone. So where was I born? I was born in Georgia, actually, in the South, Carrollton, Georgia. And that's where I went to high school, Carrollton High School. And then I went to undergrad at a college in Georgia called Mercer University, and I did biochemistry and molecular biology there. Then I wanted to see the world, so I took an adventure to Thailand for 1 year, where I taught English in a rural village in Thailand. And that experience, coupled with my love for science, not that I did any science in Thailand, but just knowing what is out there in the world, and being able to really get a sense for adventure and do something different as opposed to education, that made me apply to graduate school. And so that's how I ended up at the University of Wisconsin. So I am in my 5th year of a PhD program here at the University of Wisconsin-Madison. So still working on it, but it's been great. I mean, I think coming from the tropical Thailand, also Georgia was pretty hot, so coming here was a bit of a shock in the winters, but I have adjusted and it's been really fun.
Very good. There's a lot to be said for ice fishing. Like, I'm done with ice fishing. I'm going to we'll go on. So if you've been here the last 2 or 3 winters, you've been through a lot. So
yes,
very good. And then Professor Sugden, I don't know if you'd like to say hello. And Rebecca is in your lab, and I just wanted to say thank you for suggesting that Rebecca give the talk here at Wednesday night at the lab. And
well, for us, it's actually a privilege to be able to share with you all what we're doing, and in particular what Rebecca's doing. So thank you. You're
welcome, and thank you. It's great to get your email offering to do this, and I appreciate all the work that went into making this happen. So tonight we're going to do a little bit different than what we've done in the past. Rebecca already gave her talk with the high-end equipment from PBS Wisconsin, and the folks from PBS Wisconsin let me download that. And so we're going to run the talk, and then when that's done, Rebecca will field questions, and you can ask questions and make comments. But since it's a recorded talk, you won't be able to stop her and say anything until after the Q&A, so it's not a live presentation that way. You can try to stop her, but it won't work. So let me see how I'm going to do this. I'm going to share my screen, and I think if everybody can, once it gets rolling, if you'd be so kind as to turn off your stop your video and mute yourself. I think I'm going to do that also. We can get rolling here and see how this works. So here's this. And I'm going to hit the go button. Well, let's see, what do I need to do here? I'm going to try this and see if that'll work all right for us. Welcome everyone to Wednesday Night at the Lab. I'm Tom Zinman. I work at the Biotechnology Center at UW-Madison. I also work for the Division of Extension and for Wisconsin 4-H. And on behalf of those folks and our other co-organizers, PBS Wisconsin, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night by Zoom 50 times a year. Tonight, it's my pleasure to introduce to you Rebecca Hutcheson. She's a graduate researcher at the McArdle Center for Cancer Research here at UW-Madison. She got her undergraduate degree from Mercer University in biochemistry and molecular biology. And then she came here to study the Epstein-Barr virus and its role in causing Burkitt lymphoma. She's also interested in any virus that has the potential for causing a global pandemic. And that's one reason she shifted her research recently to studying the COVID-19 virus. Tonight, she's going to speak to us about making the COVID-19 virus safe for research. Please join me in welcoming Rebecca Hutcheson to Wednesday Night at the Lab.
Thanks for that introduction, Tom. So I wanted to start today by describing the reasons that I find myself doing this research in the first place, because I never thought that I would be studying a virus that's currently causing a pandemic. So, like Tom mentioned, I am in my 5th year of graduate school at Bill Sugden's lab, and we actually study a different virus that's called Epstein-Barr virus. And about a year ago, at the beginning of the COVID-19 outbreak in the United States, our lab collectively decided that we had the expertise to be able to make a contribution to the scientific community. And so, even though the virus that the Sugden lab studies is quite different from the coronavirus that causes COVID-19, we know that the experiments that are used to study viruses in the lab are pretty similar. And so we said to each other, hey, we really know a lot about this realm of virology, and we can help from a research standpoint. And virology is just the study of viruses. So we happen to be experts in that area, especially in the terms of molecular biology research. And so that's what we're doing as a lab. We are using our skills to contribute to this public health crisis. And the second reason that I am finding myself doing this research is actually kind of personal. And so I was a pretty weird kid, I guess, and I was captivated by viruses. And I was about 10 or so, I was super excited because I had received these giant coffee table books about disease and viruses. And I remember just poring over the pages, captivated with, these microscopic entities and how they affect humankind. And so my fascination with viruses has really stuck with me through my education until now. And, the viruses that are listed in the book, they don't include the virus that causes COVID-19 disease. And so to even be able to contribute to this field of knowledge that's, very novel, and we don't know a lot about it historically, it's very humbling to be able to add to the field of knowledge that is currently being built. And so myself and my lab mates are doing that right now. And so I want to describe today the project that we're working on. And that project is trying to really make the virus that causes COVID-19 safer for researchers to study in the lab. But before I get into that project, I would like to introduce the lab, who have been in and out for the last 8 months. And these are the members of the Sugden lab who are working right now on the COVID-19 project. So we have Roop, Mitch, myself, Tejas, Quincy, and Bill. And Bill is our principal investigator. And so now as you listen to me, describe the research that we're doing, you now have faces to put to the work that's being done. And I think that's really important. And so I wanted to recognize everyone right up front so that you know that even though you're seeing my face today, you have everyone else in your heads that has been also working on this project. And so I want to thank them too for allowing me to present this work, for all of us today. So as I mentioned, the research that we're doing involves making the coronavirus that causes COVID-19 safer for researchers to work with in the lab setting. And before I get into the details of that project, I want to kind of go over some background for 3 key things that we need everybody to know. So number 1 is the virus that causes COVID-19, which is called Sars Cov2. And then we'll talk number 2 about that virus's life cycle. And then number 3, we will talk about the facilities where that virus is currently studied. So the first thing I want to introduce is that virus that causes COVID-19. It's called Sars Cov2, and we're going to break down the name because it is a long name, but it just stands for SARS stands for Severe Acute Respiratory Syndrome because it's just a respiratory illness. Coronavirus because that's the type of virus that the virus is. It's the virus family. And the number 2, because there was a very similar virus that caused a much smaller outbreak in 2003 that was given the name SARS-CoV. So in general, most viruses are made up of 2 parts. We can think about the outer shell and the innards. And this outer shell of SARS-CoV-2 is made up of different proteins, one of which is called spike. And these spike proteins are actually common to all coronaviruses, and they actually gave the virus family its name because the word coronavirus is derived from the Latin word corona that actually means crown. And so these spike proteins are like the spikes on a crown, and they allow for the virus to enter and infect human cells. And it's actually, the spike protein is what many of the current vaccines are targeted against. So spike and the other proteins, that orange colored membrane protein that you're seeing and the yellow colored envelope protein make up the outer portion of the virus that we're going to collectively call the shell for our purposes here today. And the shell, inside the shell is the virus's genetic information. So the virus genome is made of RNA, and it's just another type of genetic material, just like humans, our genetic material is DNA. And just like humans have genes that code for things in our DNA, like what eye color you have or whether you are lactose intolerant, the virus's RNA genome has its own genes, and those are going to become important for us to talk about a little later. And so I'm going to use the word genome a lot, and I want to make sure that everybody knows what I'm talking about when I say the word genome, so I'll define it for you. The genome is just the instructions to make all the components of a living thing, whether that living thing is a person or a virus. The genome can be made of both DNA or RNA, and you can kind of think of it like a cookbook. So it's instructions to make different genes or recipes, and you can think of the genes as recipes. And the cell is going to read these recipes or these genes, and it's going to make whatever the instructions say. And so with that being said, let's move on to the second thing, which is the life cycle of this virus. So the SARS-CoV-2 life cycle sort of starts out like this. Since the virus actually can't make more of itself, like actually all viruses can't make more of themselves, they need a host to replicate. And so I'm going to use the example of a human lung cell here, and I just want everyone to know that this diagram is absolutely not to scale. So the virus attaches to the cell using that spike protein, much like the way that a key would fit into a lock. And doing that allows the virus to break into the cell and release its genome, which is that viral RNA. Okay, and this foreign RNA can't really be distinguished by the cell's machinery. So the cell actually begins to read the virus's genes and to make the proteins that make up that outer shell. And that's kind of happening on the top of the diagram. The virus's newly made proteins can then come together with the newly copied viral RNA. And that's happening simultaneously, kind of in the middle there of that diagram. And so now all that's left is for that viral RNA to come together with those outer shell proteins, and they're going to assemble into new virus particles, and then the newly made virus can leave the cell. So the simple cartoon that I showed you is one virus particle infecting one cell and releasing one new virus particle. But in reality, what happens is it's more like on the order of 1,000 new virus particles are released from an infected cell. And all of that chaos causes the body to call on the help of the immune system. And it's actually one of the reasons that humans can get so sick from SARS-CoV-2 infections. And so as researchers, we're studying this virus to help develop vaccines and treatments to treat people with COVID-19. But the researchers also have to protect themselves in the lab from infections that may occur. And so that's going to bring me to my third point, which is the facilities that SARS-CoV-2 is being studied in. And so you can categorize research labs into different biosafety levels. And these levels are in place to regulate what kind of organisms that scientists can work with in a lab facility. And they are designed not only to protect us, the laboratory personnel, but also to protect the surrounding environment and the communities. And these levels are ranked 1 through 4, and 1 is the lowest, and 4 is the highest and the most stringent. And I want to run through these pretty quickly so that everybody kind of knows what's going on at each level. So at biosafety level 1, scientists can work actually directly on a benchtop with microbes that cause mild disease, things like yeast or E. coli. At level 2, there are more precautions in place for scientists who are working with microbes that cause mild disease in humans or pathogens that don't spread through the air. And those examples include the viruses that cause hepatitis, the parasite that can cause malaria, and also things like salmonella bacteria. And so the next level up, level 3, is for research involving microbes that cause serious disease, and they can be infectious through inhalation. So examples of level 3 pathogens would include yellow fever virus, HIV, and the current plague, SARS-CoV-2. And finally, biosafety level 4 is reserved for only the most dangerous agents, those that cause severe or even fatal disease in humans, and those that really have no cures or no treatments against them. And so examples of biosafety level 4 pathogens would be things like Ebola and Marburg, and those viruses are very dangerous. And so scientists working with those viruses and microbes have to be in those sort of specialized pressurized space suits that you see in the movies, and those are real. And they also have a whole host of other precautions that you don't see in the movies that makes these facilities incredibly rare. So there's actually only 9 biosafety level 4 facilities in the entire United States, compared to some odd thousands of biosafety level 2 facilities. And so I wanted to explain all of this because SARS-CoV-2, the virus that causes COVID-19, has to be worked with at biosafety level 3. And that is a big deal for 2 reasons. The first reason is that it's really hard to do research at this level. Scientists have to be specially trained to work with these pathogens. They have to wear all the necessary gear and the respirators while working. They have to shower to leave the lab to decontaminate. And so it's really a lot to get everybody working in the lab and get experiments going. And there's a lot of regulations surrounding samples and what you can take in and out of the lab. And so that makes it really difficult to work in this space. And the second reason that working at biosafety level 3 is such a big hassle is because the number of facilities that are able to work at this level is really rare. So for example, at the University of Wisconsin-Madison, there are probably about 500 biosafety level 2 labs, but compared to fewer than 10 biosafety level 3 labs that have been approved to work on SARS-CoV-2. And so they're actually have to be built to be able to contain the pathogens because they need specialized airflow systems, they need filtration systems, double doors, they need decontamination areas, and all of that has to be planned prior to the construction of a building. And so biosafety level 3 facilities can't just pop up when we need them to. And so these two things are limiting the amount of research that can be done right now with SARS-CoV-2 infectious virus. And as we all know, it's really important to be able to study this virus and learn more about it so that we can, develop vaccines and treatments against this virus that causes COVID-19. And so, we in the lab recognized this, as everyone has, and we said, what are we going to do? We have a lot of expertise, but what can we do as a lab to help put an end to this pandemic? So it's not feasible for anyone to try to build more biosafety level 3 labs and train more scientists to work in them. But what if we tackle the other end of the problem? So what if we tame the virus itself? So our lab's goal is to make a safer version of SARS-CoV-2 so that researchers can study the life cycle of the virus, but it's going to be able to be worked at in biosafety level 2 instead of level 3. And this would mean that lots more scientists could study SARS-CoV-2 and accelerate the rate of what we learn, the treatments that we could develop, and the patients that could potentially be saved. So from here on out, I'm going to call the safer version of SARS-CoV-2 that we're developing CoV-2 DEF, where DEF stands for defective, and I'm going to explain how it's defective in a little bit. The CoV-2 DEF virus that you see in the coming diagrams is going to be green in color, and it's different biologically from the original SARS-CoV-2 virus. So we're going to build the safer derivative so ourselves and other researchers can add to what's already being done. So it is possible to study smaller portions of the SARS-CoV-2 life cycle at biosafety level 2, and that's being done now, but that really breaks up the biology of the entire life cycle, and it can become difficult to see the bigger picture. So if you think of the virus's life cycle as an orchestra and think of each portion of that life cycle as an individual musical instrument in that orchestra. For example, we can examine a violin in detail. We can learn how it works. We could take it apart. We could see how it sounds, but we don't know what that sounds like as part of an orchestra. And so we would like to be able to study the entirety of everything together. And for example, scientists have learned a lot by studying the spike protein in isolation as a little unit by itself. And that's been really valuable, but it is impossible to study all of viral infection and the human immune response to SARS-CoV-2 or test different treatments for COVID-19 if what you're studying is a piece of something that is actually working like a biological orchestra as a whole. And so what we can do is create this safer version, which is CoV-2 DEF, that's going to allow scientists to study most of the virus life cycle altogether. So to do this, let's go back to the life cycle of SARS-CoV-2, only this time we have our safer derivative CoV-2 DEF in place. So CoV-2 DEF acts very similarly through the virus life cycle until the end. And if you look closely, I've kind of put a red X over the envelope and the membrane structural genes, those orange and the yellow bits, which indicates that those have been left out of the genome. And what that means is that when the virus is making all of its proteins to build the outer shell, 2 of the crucial instructions to build those 2 proteins are missing. And so without those 2 components, that's part of the vital proteins necessary to build the shell. And so the virus particle isn't able to assemble into new virus particles. And that means the end of the life cycle doesn't happen. And that means that it's a dead end for CoV-2 DEF. So now, if we had COVID-2-Def, scientists could study the entirety of entry and infection and replication, but now COVID-2-Def virus is going to die inside this cell. And so overall, COVID-2-Def derivative is competent for replication because the COVID-2-Def RNA genome can be copied and almost all the viral proteins can be made, but it's propagation defective, which means it's not going to spread to other cells and infect them because it's not releasing new viral particles. So in the next part of the talk, I'm going to describe how we can engineer a new virus from the original SARS-CoV-2. And the idea of developing a new derivative that's safer to work with for a difficult to work with virus in the first place is not a new idea. It's actually been done with many different viruses, including HIV, which if you remember from before, I told you is worked at biosafety level 3 when you use infectious virus. And so back in the 80s and 90s, scientists were in a very similar situation that we are all in today, which is they were trying to work with a virus that was novel and it was causing outbreaks all over the world, and it had to be worked with in biosafety level 3 because of safety concerns. So luckily in 1998, researchers were able to develop a safer derivative of HIV that was able to be worked with at biosafety level 2. And it eventually became an incredibly useful tool for experimentation and for different applications that we use in our lab kind of on a regular basis. And so it's been very valuable to not only study the virus itself and prevent outbreaks, but also for the scientific community. So another example of another safe derivative is another coronavirus that's called MERS-CoV. And MERS-CoV caused a small outbreak in 2012 in the Middle East. And we know that we can be successful with SARS-CoV-2, just like scientists were successful with MERS-CoV. And we know that they had built a safe derivative, and we can also do the same thing. And especially because they are both coronaviruses, we know that this can be done. And so these derivatives are safe, they make research easier, and they help accelerate the progress of discovery. So with this safer version, scientists are going to be able to do a lot of the assays that are now performed at biosafety level 3 and move them down to level 2. For example, one of the things you may have heard a lot about in the news are antibodies, specifically antibodies that our bodies make that work against the virus to neutralize it or to kill it. So patients that have been infected with SARS-CoV-2 and recovered have developed these little Y-shaped molecules that are called antibodies that act directly against the virus. And one of the ways to check if a patient has made antibodies to something is to take a serum sample from that patient and expose it to infectious virus in a tube inside the lab. And scientists can learn a lot about studying these neutralizing antibodies that people are making. And if we can learn enough about a neutralizing antibody, we can actually produce it in the lab and give it as a treatment to those COVID-19 patients who are actually failing to make their own antibodies. So right now, these assays have to be done at biosafety level 3 because we have to use infectious SARS-CoV-2. But COVID-2-Def can be used in the same way, but it's actually safer to work with. So another thing that would be incredibly useful for the scientific community is to use COVID-2-Def to search for an inhibitor of the viral replication step. And it can be used as a potential treatment to help those who are infected with SARS-CoV-2. And these are just 2 examples of the many applications that we could use for COVID-2-Def. But right now, all these things are currently being done either with portions of the original virus or at biosafety level 3, which again is really difficult to work in. So they could be made easier using COVID-2-Def virus. Not only that, but the number of laboratories that could work on SARS-CoV-2 research would increase because we are limited right now by the need for containment level. We need to be at level 3. And so imagine if some 1,000 more labs were able to work and study SARS-CoV-2, the research that we're doing would accelerate, and perhaps we could learn crucial information that would be needed to help stop the pandemic. So now we're going to switch gears, so hang on, and we're going to talk about how we in the Sugden lab are building the COVID-2 virus. So remember, we want to engineer the COVID-2 virus to be able to perform one round of infection on a cell, but not get out of that cell. So unfortunately for us, it'd be really cool if we could just throw together the viral proteins and the viral RNA in a tube in the lab and get the virus to come out, but we can't do that. So we need all the components, but we need a more sophisticated machine to build them. So how about we use the natural virus builder, which is the host cell. So we can supply all the components of COVID-2 to a cell, and with some manipulation of biology, we can produce our virus, and it would almost be like a little virus factory for making COVID-2. So making synthetic virus is a really common molecular biology technique that's done on a regular basis. And so it's used for a lot of different applications. And these techniques of making virus in cells is not a new technique, but the virus that we're going to produce is going to be new. So the cells that we're going to use to make COVID-2 virus are called 293 cells, and they're derived from human tissue that's been immortalized in the lab, and that means that they're going to continue to divide in the dish that we keep them in indefinitely. And so we grow them in petri dishes inside of incubators that are kept at the human body temperature, and the cells adhere to the bottom of the plastic plate, and they're supplemented with a red-colored liquid medium that supply the cells with all the necessary nutrients and growth factors that they need to grow in the dish. And so the 293 cells that I'm showing you in the image have been fluorescently colored so that you can see the nucleus of the cells in red and the cytoplasm in green. And my cartoon of the 293 cells is that blue blob that's in the dish over there. And I'm going to use the image of the 293 cells that's got cartoon from here on out so that we can kind of keep track. These cells are going to house, or they're going to be supplied with all the components that are necessary to make COVID-2 virus. So I'm going to talk about the components of COVID-2 in two parts. So I'm going to talk about the viral genome and then the structural proteins that are going to make up the outer shell of that virus. And I'll talk about them in the context of COVID-2, but I'm actually going to use those colored versions of the outer shell proteins so that we can follow them and we don't get confused with the same colors. So if you remember from earlier on, we're going to make COVID-2 virus unable to make new virus in the cells that it's infecting, but COVID-2 has to have all the components of the original. And one important point that I really want to clarify is that we're using what we know already about SARS-CoV-2, which is the original virus, to build COVID-2, but we're actually starting entirely from scratch. We have no starting material, and so we know the genome sequence of the original virus, and we can read those instructions and use that information to build new instructions so that we can make COVID-2. So with that in mind, let's delve into the first component, which is the viral genome that we're going to supply the 293 cells with. So before we get into that, though, here's a key concept that I need everyone to hang on into their brains. So in biology, the usual order of things goes like this. We have DNA that's read and made into RNA, and then that RNA is made into protein. And these proteins are what make up almost everything in the cell, and therefore our bodies, and almost every living thing on Earth. So this is the central rule of biology for our purposes today. It goes DNA, RNA, protein. So the SARS-CoV-2 genome is made of RNA, but RNA is unstable and it's actually really hard to work with in the lab when you compare it to DNA. Both RNA and DNA can be read by the cell, and the instructions are written to give the same protein products. And so we're going to actually use DNA to make the COVID-2 genome instead. And this is still going to work as the original virus would, because the DNA can be turned into RNA, and then that's going to serve as the virus genome would naturally. So we need to rewrite the new instructions for COVID-2 using the genome sequence of SARS-CoV-2. So let me describe how we're going to do that and how we've designed these new instructions. So we're going to zoom in on the genome. So instead of those sort of zigzag, the circular zigzag, I want you to think of the genome stretched out so that we can read it left to right like a sentence. And the sequence of the DNA genome is made up of 4 letters, A, T, C, and G. And certain combinations of these 4 letters are read by the cell as genes, which I'm representing as those colored blocks. There are a lot more genes and elements in the COVID-2 genome that we're building, so these elements have been simplified and grayed out so that we can focus on the key genes. Not that these grayed out bits aren't important though. They actually serve as the instructions for a lot of key proteins that the virus needs, one of which is actually copying the virus's genome. And so my simple genome that I'm showing you today of these sort of rectangles is incredibly oversimplified. We only have time to talk about a few genes today, but the virus's complexity is something to appreciate when you consider what we've designed. And so as the cell machinery reads these genes, we're going to make the DNA into RNA and then the RNA into protein from there. And so we started first by designing the sequence of COVID-2 on paper using a computer program, where we can see all the genes of the original sequence and then decide which parts we want to keep from the original instructions to write the instructions for COVID-2. So imagine that we have copied those genes into a new document. And you'll notice that we've left out the envelope and the membrane genes, those orange and the yellow colored instructions that are eventually going to make their respective proteins in the outer shell of the virus. By not including these components from the original instructions, COVID-2 that carries this genome is going to be unable to make new virus particles in the cells it infects, because it doesn't have those instructions to make these 2 proteins. Okay, so that's really key. In place of the envelope and the membrane genes, we've added back some special genes in place of the ones we got rid of. And these genes are going to give rise to proteins that are actually colored. One is the green fluorescent protein that was isolated from jellyfish, and one is the same enzyme that actually makes fireflies glow, and that's called luciferase. So by adding these genes in, we can visualize anything our COVID-2 virus infects by looking for green color and the luciferase enzyme. So another key element that we've added to this COVID-2 genome is an element that allows us to turn on and off all the elements on the entire stretch of DNA. And it's this little blue site that I'm showing you, and it's called a Tet Crab Element, and it works because it's inducible. So you can think of it like a light switch, which is usually turned off until we add a drug that's called doxycycline. And the way this works is a really cool piece of biology. So let's think of our string of DNA, but since we've included the Tet Crab element in the design, it acts like a signal to other proteins in the cell. And these other proteins actually sit on top of the DNA right in the way. And these proteins prevent the DNA from being read by the cell's reader machinery, so that none of the genes that are on this piece of DNA can be read, because the machinery has to start at the beginning of the genes. And so without doxycycline, these proteins are in the way and nothing is being made because they're sitting on the top of the DNA. But when doxycycline is added, it actually binds and removes those proteins that are in the way, and then that allows the DNA to become accessible to the cell's reading machinery, which allows for the genes to be read and for those proteins to eventually be produced. And so this is one of the ways that we can manipulate the biology of the COVID-2 genome so that it's safer to use. So overall, this means that if we have the COVID-2 genome inside 293 cells, the only way that COVID-2 virus is going to be made is if we add doxycycline to those cells. If we take away that doxycycline, the production of the COVID-2 virus is going to stop. And doxycycline is a really easy thing to work with. It can easily be added to the cells because it's a drug. So the molecule that you're seeing is actually dissolved into a liquid. And then that liquid, a small amount, can be added to the red colored liquid media that the cells live in. And the doxycycline is a molecule, so it's free floating, and it can diffuse into those cells, and it can go to the DNA, and it can then remove those proteins that are sitting in the way on that little blue site. And then the genes can then be read by the cell's machinery, and we can get the proteins that we want to produce. When we want to take away the doxycycline, all we do is just suck off the old media, and we put new fresh media on that doesn't contain the doxycycline. So now that I've described all of the genes and the elements on our Covi-2-Def genome, I want to tell you how we are physically building this lengthy molecule. So the original Sars Cov2 genome is about 30,000 letters in length. And we need to build a DNA molecule that's actually more than 40,000 letters in length. And we need to build it from scratch. And so that's a really big undertaking. So to do this, we can order fragments of synthetic DNA from companies, but those actually can only be built to about 2,000 letters in length. So we have to order those fragments and then piece them all together in the right order using a process called DNA ligation. And so if any of those fragments are incorrect, or if any of the ligation reactions introduce an error, we have to correct that error. So the cell reads the DNA sequence letters in groups of 3. And one letter change can make a really huge difference in the way the protein is produced. So for example, if you misspell the word bar to the word war, you are really changing the context of what you're trying to say. And that's true for the DNA sequence as well. So we're working really hard in the lab to have a perfectly printed DNA fragment. And I will tell you right now, this has not been an easy thing to accomplish. We are actually still working on it, which I'll tell you about a little later. But let me just describe one of the snags for you so that you kind of get an idea of how hard this has been for everyone working on it. So as I mentioned, the fragments have to be put together using ligation. But before we get to that step, all of the fragments have to individually be put into bacteria and grown up so that they can be amplified. And so we found that some of the pieces are actually poisonous to the bacteria that we're trying to grow them in. And that took us like a month to figure out. And then we had to manipulate the biology of the bacteria so that we can put our fragment in, so that we could then, grow up the fragments and use them to ligate to the others. And so, it has been a wild ride, but we are about, I would say 60% done with building this really lengthy molecule. And we're working really hard to finish it off right now. So now we're going to talk about the other part of the DNA, or the other part that we need to make Covi-2-Def virus, which are the structural proteins that make up the outer shell of the virus. And at this point, we've talked about putting the genome, the Covi-2-Def genome, into 293 cells. And if the genome is the only thing that we have in the 293 cells, and we turn on those genes using doxycycline, we're going to make the DNA into RNA, and that'll give us the RNA genome. And then it's also going to produce the proteins, and it's going to produce spike, that green fluorescent protein, luciferase, and all those other grayed out proteins that we don't have time to talk about. So the missing proteins that we need to make virus are the yellow sort of envelope and the orange membrane proteins. And since we purposefully left out their DNA in the instructions, we need to supply those two proteins to the 293 cells if we want them to make virus for us. And so to make our Covi-2-Def system safer, we're gonna give these two proteins to the 293 cells in 2 different ways. And I wanna talk about the orange protein first, that membrane protein. And so the membrane protein is actually the most abundant protein in the viral particle. So we need a lot of it to be made for all the virus particles to be made for Covi-2-Def. And to supply the 293 cells with orange membrane protein, we're going to use the same doxycycline inducible system because the cells already contain that system. The membrane gene is going to be on a different piece of DNA inside these cells, not on the Cov2-Def DNA genome. And this makes the system safer by using 2 pieces of DNA instead of 1. And so first we introduce this membrane gene directly into the cells using a process called retroviral transduction. So remember, these 293 cells have their own DNA. They have 46 human chromosomes because they are human cells. And so we were able to integrate this piece of DNA into the 293 cell DNA. And so on this piece of DNA is also the blue site that allows for induction using doxycycline. And so when doxycycline is added to these cells, the light switch is flipped on, and then we get membrane protein made. And for simplicity, I have been calling the membrane protein orange, but in reality, it's actually just clear. It doesn't have a color. And so that we could follow them in the cells, we actually included a fluorescent protein just after the membrane protein that's called M-cherry. And M-cherry is going to be red. And so in the presence of doxycycline, we're going to make M-cherry protein as well as that membrane protein. So having the M-cherry component in the cells allows us to quickly check whether our doxycycline system is working. So I'm going to show you 2 images of the 293 cells that contain the integrated DNA piece that encodes the membrane gene and encodes the M-cherry gene. Both of the photos of the 293 cells on the left are containing the integrated DNA piece. But on the top panel, the doxycycline has been added to those cells. And so you can see that those cells turn red because they're making the M-cherry protein, which also means that they're making the membrane protein. And through this test, we know that our cells can be successfully making the membrane protein that we want. We do have other ways to tell if the protein is being made in these cells, but this is the most visual way to see that, and it's the easiest way to show you here today. So now the final piece of the puzzle, or in this case, the Cov2-Def viral particle, is the envelope protein that's yellow in this case. And this protein is going to be provided through direct RNA transfection. And so what that means is that we make the 293 cells take up RNA directly. And from there, it can be translated into the envelope protein. So, of course, we couldn't just snap our fingers and make this molecule. We had to build this molecule by taking a DNA molecule and introducing that into cells, having the cells then produce RNA, and then we had to purify that RNA piece out of those cells and put it in a tube, and then only then we could introduce this RNA molecule into the 293 cells and have them produce our envelope protein. So once we've done that, the RNA is actually used by the cell and it's degraded, meaning that it's not going to stick around and accumulate in these cells. So at this point, you may be wondering why we've kind of made life really difficult for ourselves, seemingly, because we have many different methods for supplying these cells with the components to make Cov2-Def, but it was actually intentional. So by using both DNA and RNA on different molecules, it actually makes the system very safe because there's no way that we could somehow combine these components together to make the original SARS-CoV-2 virus. Biologically, it's just not possible for these things to come together to make the original. And that's really important, especially because we want our system to be really safe for researchers to use. And this is true because we're using both DNA and RNA. They're on different molecules, but also 2 of the elements are inducible with doxycycline, meaning that we're not going to make anything in these cells unless we give the cells doxycycline, which we can control. So now I've described all the pieces for you, so let's put it all together. We have the Cov2-Def DNA genome, which is a really long piece of DNA that contains all the instructions to make virus, except for those orange and those yellow genes. So because the virus cannot be made without those 2 proteins, we're going to supply them in two different ways. The membrane through a DNA that's been integrated into the 293 cells, and then the envelope through direct delivery of RNA into the cells. And so we only supply that yellow envelope RNA when we're ready to make virus. And then we're going to add doxycycline to the cells, which is going to cause the switch to flip on. And then we make orange protein. We're going to make all the proteins encoded on the Covi-2-def DNA genome. And then we're also going to copy that DNA genome into RNA. And so now that RNA genome, which is the natural state, can be encapsulated by the outer shell proteins, and then voila, we have our Covi-2-def virus, and that's going to be secreted into the media of those cells. And so then we can actually just collect that media and put it into a tube, and we have our Covi-2-def virus produced. And this virus can now be used to infect other cells by putting the media, which has the virus inside of it, directly onto new cells. And if these new cells are able to be infected, Covi-2-def virus is going to enter, it's going to uncoat, and the RNA genome inside is going to provide the instructions to make all the proteins, except for the orange and the yellow membrane and envelope. And instead, that's going to make the green fluorescent protein and the firefly luciferase. So in this way, we can actually visualize which cells are infected with Covi-2-def because we see the green fluorescent protein and the luciferase. And that's a really useful tool for scientists to use when performing different assays, because now there's actually a direct readout in color, which can be measured with instruments or microscopes. And so here's an example of cells that have been infected with another coronavirus that we work with in the lab. And this virus is called NL63, and it's another coronavirus that is much more benign, and it only causes mild colds in humans, or it causes nothing at all. And so we're using NL63 as a model to study infection in this case. And we can't easily tell from the image on the left that's using bright field light which cells are infected with the virus. The picture on the right is the same picture, it's the same cells, but now you're looking at it through a fluorescence microscope, which tells us that the cells are making green fluorescent protein and thus are infected with the virus. And this is going to let us study the cells as they grow, and we can monitor the infection of these green cells. So as we come to the end, I want to describe what we've accomplished so far. And I have tried to lay all of this out for you so that it's pretty straightforward, such that, this protein is going to do this, and this DNA does this, and we put this piece here. But in reality, biology is quite messy, and usually the first time you try something, it's not going to work. And most likely the 15th time you try something, it's not going to work. And so the most challenging piece of this project by far has been building that Covi-2-def DNA genome from those fragments that I described. And we're about 60% done with that guy. And so then the orange membrane and the yellow envelope proteins, getting those to be made inside of the cells with their respective systems, are both at about the same level of completion, probably about 85%. And right now what we're doing is we're testing those components to make sure that those proteins can come together with the other proteins in the outer shell and make empty virus particles. And that just means that we aren't putting any of the genome inside, we're just kind of creating this empty shell, and we're optimizing the steps that we're taking to create those shells so that when we do eventually stick the genome in our system, we have the perfect conditions and the right settings so that we can make a lot of virus when we're ready to do so. So all of these things are currently being worked at biosafety level 2, and they're being worked at separately. But whenever we're ready to put all of them together, we're actually going to move to biosafety level 3, and we're going to test that the Covi-2-def virus that's produced from these cells is not able to infect cells for more than one round, and that those virus particles that get in stay inside that cell and they die inside that cell. And so only then are we going to be ready to share this research and these components with the world so that more research can be done on SARS-CoV-2. So overall, we have designed a system to help accelerate research by allowing more to be done at a biosafety level 2 instead of 3. It's a system that is going to protect scientists from having to work with the original SARS-CoV-2 virus, and it's a system that can be handled by researchers to be safer biologically. And so COVID-2-Def can provide a faster, safer solution to research on SARS-CoV-2, which hopefully will help to end the COVID-19 pandemic and accelerate the rate at what we learn. So with that, I'd like to close by giving a huge thank you to the University of Wisconsin-Madison, especially the Office of the Vice Chancellor for Research and Graduate Education and the McArdle Laboratory for Cancer Research for their support. And we want to thank our colleagues who have been working really closely with us to get this project off the ground. And so I want to say thank you to them. I also want to thank the National Cancer Institute and Promega for their support. Of course, I want to thank Wednesday Night at the Lab and Wisconsin PBS and all of those organizers for helping me prepare and record this talk and for the opportunity really to present here today. And so I sincerely hope that I have taught you a little bit about molecular biology, a little bit about SARS-CoV-2, and of course about the COVID-2-Def derivative that's going to be safer that we're building right now. And also why it matters, why it's important to do so. So in the Sugden Lab, we're working really hard right now. We are day in and day out in the lab, and we're hoping to complete the COVID-2-Def project in the coming months so that we could share this research and share these components with other people who would like to do COVID-19 research but can't because of containment requirements. And so we're really excited about this work. I can't stress that enough. If everyone were here, they would say the same thing. And we know it can make a real difference in helping us learn about SARS-CoV-2 and potentially help to end the pandemic. And so now I want to thank you for listening. Thanks.
All right, everyone. Thank you very much, Rebecca. If you can come back on and we'll take some questions from folks. I think people can unmute themselves if they like. They can also type questions in the chat. And anything else you'd like to offer? Let's see. So the participants, I've got that checked that you can unmute yourself. So if anyone would like to unmute, Darius, would you like to start off?
I'm wondering about the envelope proteins in the SARS-CoV-2-Def, how they differ from the envelope proteins of the original infectious SARS-CoV-2. You need some sort of envelope protein to get that whole virus particle assembled, but you want a disabled one.
Right.
I'm a little curious there. That's not clear to me.
Okay, sure. Great question. So they are actually almost the exact same as the original SARS-CoV-2 virus, the envelope that we're providing to those cells, except for that they have been codon optimized, which just means it's a little easier for the cells to make. But I think the key thing is that even though they are essentially the same protein that SARS-CoV-2 has, and then COVID-2-Def is provided, what you get is that virus COVID-2-Def that is lacking the instructions of the envelope and the membrane, so that when it infects new cells, it can make everything it wants to, other than the envelope and the membrane. So we can get infection in one cell or multiple cells, but then that infection stops in those cells. It can't spread to surrounding cells. And that's one of the big reasons that safety is such an issue, because if you're wanting to study the life cycle and study Sars Cov2, you have to propagate the virus. You kind of have to, when you infect, you have more cells and you don't want it to spread. And so this system, by just not including that envelope instruction, you don't get spread of infection in an infected cell. Does that answer your question?
Yes, I think so. Thank you.
Sure.
And
Yeah,
from the
I have
chat-
a question. Are you going to produce a series of different spike proteins representing the variants that are beginning to crop up?
We thought we might get that question. Thank you so much for asking it. I was really excited. I hope somebody would ask. So yeah, that's a huge thing right now, is that we have so many spike variants in community spread that would be interesting to study. And one of the ways that we could do that is the Covi-2-Def genome that we have built is we've designed it such that we can easily change and modify the spike protein. So right now the spike protein in the Covi-2-Def genome is wild type, but it can easily be changed because of the way we've designed the system itself. So yes, it is not, we're not studying variants now, but it could easily be done. So yeah, great question.
Thank you.
And then from the chat, James asks, what is the half-life of the envelope protein RNA in the transfections?
So I'm gonna give an answer that's slightly, well, the answer is we don't know. We could measure it. It's probably on the order of, so let's see, if we are transfecting the RNA in, it's gonna probably be on the order of a day. We know that we can produce protein from those cells that have taken up that RNA, but it hasn't been measured directly. And so I'm just going to leave it at that, but it's not going to be, weeks. It'll be on the order of, 12 hours, 24 hours. And then that'll be produced into protein, or it'll be degraded by the cells.
Great. Another question from the audience, someone who'd like to speak. We've got one more in the chat, but if I'll tend to go back and forth, one from the spoken audience and one from the chat, if anybody's got one. If not, I'll go to the chat with Raj Gopal. Did you do research on developing COVID-Def at different ambient temperatures? Since with COVID-19, the level of ambient temperature can render the virus ineffective.
Great question. So we haven't gotten that far yet into testing the temperature that's optimal for Covi-2-Def, but what we do know is by studying NL63, which is another coronavirus that causes very benign disease or no disease at all. So Bill in the lab has been working a lot with NL63, and he has found that NL63 is temperature sensitive, meaning that most cells are grown at 37 degrees Celsius, but NL63 actually prefers a little colder. So it would be something that we would look for once we produce Covi-2-Def.
Very good. Other questions, either in the chat or directly? While you're thinking of other questions, I'd be interested in knowing how it looked and felt to you, the idea of running the recorded talk through the Zoom, and whether that was satisfactory or how that all looked, because tonight, as Professor Sugden pointed out, for us, this was an experiment, and I wouldn't mind getting to hear the pros and cons of what you think about how it worked or didn't work very well. I was very pleased with how it looked on mine. I'm not sure how it looked on through the Zoom and on the YouTube video.
Yeah, it looked really great on my end. It was nice. I was able to have a snack while I watched myself, so that was fun.
And Paul Lambert and a couple other people reminded me my video was on, and it was on because I forgot to turn it off, and I wasn't even sure if I turned it off, if you could screen share, which shows how rarely I screen share. So I just got to be here and hopefully was watching okay. Here we go from Lawrence Winkler. At the time of your recording, you believed you were at about 60%. Where are you today?
Good question. So as I mentioned before, research is a sort of whirlwind of ups and downs. I would say that we are with the Covi-2-Def molecule. So that means, the somewhat 40,000 bases that we have to build from scratch. We're probably about 80% done with that right now. There are some snags in terms of some of the fragments are really unstable. And so that trying to move from, a molecule that is small to put it into a molecule that's much larger and has to be stable and has to be expressed and prepped up and grown so that we can use it, I would say we're about 80% there. In terms of supplying the different Covi-2-Def proteins, those outer shell proteins, to the system, we've had to sort of swivel and change what we were originally intending. And that is because of the amount of protein that we're getting out of those 293 cells. And so it really has been sort of a give and take. It seems like some of the project was working and those people are excited. And then some of the project is really sluggish and we're all, still building it together. But I would hope that we can kind of finish it off in the next month or two.
Thank you. Melissa Hutchison is asking, I'm going to ask first, who's Melissa?
Melissa is my twin sister. She is, yep, I have a twin sister. She is a PhD student at the University of Michigan, and she is studying particle physics, which I know nothing about. So it'll be interesting to see her question.
Well, she says, which part of the building of the Covi-Def are you working on? What are you doing right now?
Oh, okay. So right now, my main portion of the project has been supplying that membrane protein to the cells. And so we have some people who are building the molecule, that DNA molecule that's very massive and difficult. We have some people working on the structural components, and my portion is to work with the membrane protein, express it in cells, and to test whether it comes together nicely and plays nicely with all the other components. So that's what I'm currently working on.
And when we get closer to being done, if Melissa's willing to come on live and tell us about her research, that'd be pretty cool too.
That would be
cool. Every
cool.
day you get two people for one. Sandy Rotter's asking, do you know of others working on the same type of project?
Yes, I don't know them personally, obviously, but there have been some recent small publications that have come out where it's a similar system in the sense that you have a version of SARS-CoV-2 that is a little bit safer to handle in biosafety level 2, or its components are broken down such that it's easier to research portions. But there has yet to be a mammalian cell system such that SARS-CoV-2 or a modified version can be produced with and worked at biosafety level 2. So yeah, it's kind of a race to the finish line, if you will, for us to finish this up and get it going and share it with others. So, I mean, I think that anything that is being done with this virus is really valuable. So we don't really care if we're first, it's not a race to the finish line in that, we won, we beat everybody. It's just, we want everyone to be able to study this virus in a safe way. And so if we can get our system up and running, which we think we can, that'll be perfect. And we'll share it with whoever wants it. But if other people do it before us, they will likely do it in a different way because there's so many different ways to put this thing together. But yeah, whoever finishes and whoever does it is going to be super valuable to the scientific community.
Thank you. Jean and Russ ask, when you make the COVID-def cell or virus, how will you share it with the other labs?
Good question. So sharing biological material is a really frequent thing that's done in the research community. It depends on what you're sharing. So when we get things from other labs or other companies, it depends what it is and how it comes. So it can ship by FedEx, it can ship by UPS. But for example, cells, live cells are frozen down, and those are frozen and stored on dry ice, and those can be received in the mail. So for our system, what we would likely do is have this cell line that can be frozen down so that it's stable, and then it would be shipped on dry ice with all the other components to different labs who would want to work with it. And then it's really cool. You can actually sort of just thaw those cell lines. They can be thawed at room temperature, and then they go to the human body temperature, which is 37°C, and they continue to grow. So you can kind of freeze them down like cryopreservation, and then you can bring them back to life. So it can be shared worldwide. You have to have a fast shipping company so the ice doesn't melt, but it's a very possible thing to do, and that's how we get a lot of our reagents.
Very good. Other questions? From Amy, she asks, what's one thing you wish the general non-scientific public knew about COVID-19?
For me personally, I wish that the scientific method was more understood. So, you've heard me talk today about a project that we're working on, and it's not that we know everything from the start. We don't. We're very clueless. Like, we're as clueless as everyone else, other than the fact that we have some skills and some knowledge that will be able to accomplish these things. But we don't have all the answers yet. We are actively working to find those answers. But for the public to just understand that, in the beginning of the pandemic, people were saying that you could do this or you couldn't do this, and then they changed their mind and said you could do this. It's not that we changed our minds for the fun of it. We learned new information. And so I think that's one of the most important things that I would love the public to understand, because new things are being learned every day. The frustrations that the public feels are totally valid. Like, I would understand if things are changing and the rules are changing, but it's because we're learning new information, and that is the scientific method. So it would be great to have that a better understanding for everyone.
Yeah, I've been working on that for 29 and a half years.
I know, all right.
Got another 29 and a half to go. Thank you for that. I'd like to ask Professor Sugden, and you could chime in also, but you mentioned that you completely changed the gist of your research about a year ago. How does that work from the point of view of your funders? Did you have to get permission to do that? Do you say this is a national emergency and this is what we're going to do, and let your funders know? I'm just wondering how that worked for you specifically and for other labs in McCardle and other labs on campus and any other labs listening in.
So shall I do it, or Rebecca, would you like to answer? Okay, I'll do it. Okay, so first of all, the federal government, NCI, which is the National Cancer Institute, volunteered to provide supplemental funding for any of us who were going to work on COVID-19. So I'm funded we're funded to work on Epstein-Barr virus by the National Cancer Institute, but they recognized that they wanted to be able to direct those or help those who are funded to do other research to work on this problem. Also locally, the university through the Vice Chancellor's office has provided us funding. Promega has helped provide equipment for BSL-3. So there's been a lot of ways that people have provided support for us, we actually are still working on some of the problems that we were working on prior to the pandemic, but there have been tremendous support financially. And I mean, we could always use a little bit more, but in fact, I think we've been very well supported to work on something that was distinct from what we had initially grant money to work on, because people recognize the need. Does that answer?
Yeah, and it's not anything like has happened in my professional career that there would be something where people, with the possible exception of people who were doing bioterrorism research after 9/11 and the October 2001 bioterrorism attacks, but to be able to say, I'm going to stop doing what I was doing and that I was agreed to be funded to do, and I'm going to do other stuff. And it's just very interesting that people encouraged it. They wanted you to go ahead and do it. They gave you extra money.
They did, and very rapidly, the university gave us sort of call it an exceptional status that we could come in and work. So we've been working in the lab since May 1st. And that too was recognized as being important by the university, because most of the people were excluded from working in the labs.
March 13th on is my memory. Is that about right?
Yeah. But as of May 1st, all of us in the lab were allowed to come and work, and we worked in shifts so we didn't infect each other were anybody to be infected. But so the university's been superb too.
Excellent. Thank you. Did you want to add anything, Rebecca?
No, just that I feel really lucky to be able to sort of have the opportunity. I know that sort of sounds like, oh, what I would normally say, but I actually do feel lucky. I mean, the virus that we study causes cancer in certain populations, very rarely, but I have been studying that virus for 4 years. And, to be able to say like, hey, there's this new virus, it's infecting everyone, that's something that I never imagined that I would have the skills to do at this time in my career. And as a young kid, I sort of thought, well, I remember the Ebola outbreaks. I was in college, and I remember thinking, man, if I could just contribute somehow, that'd be so cool, because I was learning all this biology and I thought it was really cool. So to be now, I mean, the pandemic has been so damaging for everyone in this world, but the fact that I am able to contribute with my lab, we're all working together. It's been really collaborative. It's been a whole new area of research that I knew nothing about, and the lab really kind of took on as our project. But that has been something for me, for the university, and for us to write some of these grants and these supplemental grants to get funding to work on something different. I'm still working on my old project, and that's good, but to do something new, I think it makes me a better scientist. And as I continue to train, I hope that this experience, not only is like a weird time that we look back on, but something that I can really value. And I think that will happen. So just say
Great.
that.
A couple more from the chat. Everybody can read these. Raj Gopal asked, last Sunday on Face the Nation, it was reported the Chinese military was testing the virus with animals, and it leaked and made the virology staff ill. Do you think that they were not operating at biosafety level 4?
I don't know. I did not see that information. But I think it would be good to get more details on that. But what I will say is that, the systems that we have in place in this country and many other countries have in place are there for a reason. And so I think that the way that we work tends to be very, very regulated. I would not say that it's been easy to do any of this research. We've had to jump through tons of hoops with the biosafety department and get a lot of things signed off on to be able to work with. And so, I don't want to be able to speak for what happened in that instance, but I can tell you that typically the regulations behind taking a virus that can be so dangerous for humans to be infected with, that's not taken lightly, especially in this country. So I'll just say that.
Nicely done.
Thank you.
Raj, you got to go easier on people on those questions.
Oh, I think it's a
Sandy-
valid question, but you know.
Well, it was. Sandy asks, how much longer will the change to Rebecca's thesis impact the time to completion of her PhD dissertation? He notes, we all appreciate the added time.
Yeah, so this is not a usual thing that happens when a PhD student sets out to kind of defend and do the thesis work. I think, I'm going to say this optimistically, I don't think it's going to add any extra time because right now I'm in a position where I have about 9 or 10 months more of experiments to do on my own personal project. And one of the really cool things for me personally that I found is that I tend to work in bursts, but I also have a lot of motivation and a lot of energy when I have too many things to do. So I have been working on my own project, on the time that I am not working on our COVID-2 project. And that's worked really well for me. I think the other thing is that I would just be at home sort of twiddling my thumbs if I wasn't in the lab. And so the fact that I can still go into the lab and see the people, we're wearing masks, we're very careful, we social distance, but we can see each other every day. I have a very regulated routine that gets me out of bed and gets me going in the morning. And I don't have anything other social things going on in the evening. So to be in the lab is really a solace right now. And I'm hoping that that keeps me on track for graduation. But that's where I am, and I'm very grateful. And I say that honestly, like I'm really grateful.
Yeah. So a lot of people couldn't get into the labs, and as you pointed out, that leaves you wondering, what am I going to do and how do I keep making progress towards graduation? So this is good news. Would Melissa, your sister, like to come on and say hello? Because I think we're close to the end of the lines of questions.
She might.
Hi.
Hi, how's it going?
Hey, this is Melissa. I'm not going to turn on my video because you can just look at Rebecca. We look the same. But yes, hi. I didn't realize that if I asked a question in the chat, my name would be shown. But yes, I'm Rebecca's twin sister, and I'm working on a PhD in particle physics at University of Michigan.
Which particles are you working on?
I'm studying a particle called the kaon. And we're trying to measure something that's 1 in 30 billion. So we have to make a bunch of kaons, and then they're unstable particles, and after a certain amount of time they decay, and we're looking for this one really rare decay that can hopefully tell us something about the matter and antimatter asymmetry in the universe. So not saving the world stuff like Rebecca's doing, but important research in and of itself.
So what was dinner like at your at your table in high school?
I think it was a lot of like, did you finish the organic chemistry homework? Because I didn't get that one, or like, it was not this advanced. I think we were just kind of normal kids struggling.
Very impressive. Way to go. Thank you, Melissa, for coming on. Thanks for asking the question. Thanks for sharing. And I've never heard of kaons before, so I'm one more subatomic particle I've heard of, so that's good. Probably 200 more to go. Any other questions? Oh, here's one more new message. Let's see. Helen asks, have Chinese scientists shared any significant findings with American scientists?
So one of the great things about research is that whenever you have findings that, you think that should be published, there are these journals that are preprints. And so if you have data that you believe to be valuable to the scientific community, usually what happens if you want to publish a paper is that that paper goes under review and it's peer reviewed so that multiple colleagues will read it and say whether it is rigorous enough to be published. But one of the things that has become really popular in the last year is these preprint journals, and those are not peer reviewed. So you sort of, when you're reading through them, you have to be a little more critical and a little bit more like, hey, how did they do their studies? Are they rigorous? But all of that can be shared quickly because the peer review process can take anywhere from months to years. And so if you have this information and you said, hey, we did a study, we want to share information. If you want to publish in a journal, that can take a while. And that's just how it works. But with these preprints, you can get information out a lot faster. And there are multiple of these, and we check them, and people are publishing in them, and it's not something that is a permanent fix. Like once you have data, it's most likely going to be published in a peer-reviewed journal. But to get the news out faster, these preprints exist. And so I say all that because whoever is doing research on SARS-CoV-2, we don't know who's holding back information because we don't know the labs that are doing that research. But what we do know is that countries from all over the world, China, everywhere included, is publishing in these preprints. They're publishing what they can in peer-reviewed journals. And so what we're seeing is that people are being really generous with what they've done, and they are sharing their information. And what we need to do as scientists is make sure that when we read these papers, that they are rigorous and that we can say, hey, that makes sense. They did these experiments and we know how those experiments technically work. What they conclude is this. Do we believe that? And so I can't say anything for people who may be doing science in their basements, but from what I can say is that, what is being published is coming from all over the world, China included.
Great, thank you. Bia has a question. Bia, do you want to go ahead and speak your question?
Yeah, I will. My question is, are COVID-19 researchers considered medical professionals in terms of getting the vaccine, or is working with the Covi deaf safe enough that you're in the list with everyone else?
Great question. So I have not received the vaccine, and that is because it is safe enough to be able to work with and not be considered essential personnel. So Bill, I'm sorry, Bill, you are very old, and Bill has received the vaccine because he is a member of that community. But there are also 2 members in our lab who are working in biosafety level 3. And so they are considered working with infectious SARS-CoV-2. You are eligible to receive the vaccine. So they have received the vaccine too. So yeah, if you're working at BSL 3 and you're dealing with live samples, vaccinated. If you're not, we're part of a population that will likely be vaccinated soon, but yeah, we are not considered essential worker personnel like healthcare workers.
Thank you.
Awesome, thank you.
Raj asks, did you make vaccine manufacturers aware of your work, and did they express funding interest?
Bill may be able to answer this one more. To my knowledge, we have not made them aware, probably because we're not there yet. But as Raj is suggesting, this COVID-2 deaf version of the SARS-CoV-2, it technically would be a very strong vaccine candidate. But we are a long way away from, getting this to be a successful candidate in terms of a vaccine. But to my knowledge, that has not been something that we've planned on doing, mostly because this is for research purposes and not to take to pharmaceutical.
Very good. Thanks, Mike Rip, for your generous comment there, and I agree. From James Line-Weber, we've had the question is, where is your BSL-3 lab?
So the BSL-3 lab is in the veterinary medicine building on campus. And it is in collaboration with a researcher named Jorge Osorio. And so he has a lab and he studies a number of different viruses, and they have begun working on some Sars Cov2 research. So we are sort of sharing a space, but it's in the vet med building, and it is up and running. And yeah.
Good. Yeah, thank you. I think it's at 8:30, and that was a pretty good series of questions. I really appreciate it. Not seeing any other in the chat, and if anybody'd like to chime in, that'd be great. If not, thank you very much, Rebecca. Thanks for the time and preparing. The extra time it takes to record with PBS Wisconsin, you got to do a 1 hour check on a Tuesday, and then you got to come back and do the recording on the following Wednesday. It's more of a time commitment than in the old days when we just got together here at the auditorium and you'd come in at 6:45 and give your talk from 7 to 8 and answer questions from 8 to 8:30, 8:15, and then linger at the lectern. So I really appreciate this. Professor Sugden, thank you very much again for suggesting this, for making it happen. For everybody else who came on today, really appreciate it. And if you have any suggestions, my email address is my last name, Zinnan, at biotech, biotech.wisc.edu. And like I said, this was an experiment for us tonight. We have to figure out how to do it again better, and see how it goes. I might even learn how to turn off my video while I'm screen sharing. Anything else, Bia?
No, I just love all the support in the chat that's coming in right now.
Yeah, that's really
Thank
great.
you, everyone.
And it says love from Mom, so that's doing good there. You
Yes.
got your family here supporting you.
Yeah.
And before I go, I do want to say thanks to Bia. She's she makes sure all the stuff works as well as it does. Neither she or I are particularly technological geniuses, but if it was just me, it would have been one crash after another. And so I really appreciate it, Bia. So thanks again, Rebecca. Thank you again, Professor Sugden. Hope to see you next week. We're going to have a talk on wolf ecology. And since we just have the wolf hunt go way past its quota today, it's going to be very interesting to hear next week what Professor Adrian Trevis has to say. Talk to everybody soon, and thanks.
Thanks.
Thank
Thank
you.
you, guys.
Great job.
Yep. It was.
Thank you.
Great job.
So if Rebecca's mom is still here, do you want to come on and say hi to your two daughters?
Did she leave yet? No, she's still here.
All right. She ought to get some credit for this. Man. And B, have we ever had two people come on that were, like not only just siblings, but twins? And
I didn't know we haven't, but I think that's super impressive. Both PhD, that's amazing.
yeah.
Well, hello, everybody. This is Karen Graves, Rebecca and Melissa's mother. And it took me a minute to figure out how to turn off my microphone or turn on my microphone. Yeah, the girls, ever since they were really, really small, we had a rule in our house that there was only one store that we could go to. And it didn't really, there was no cap, but it was the bookstore. And if you went to the bookstore, you could basically get everything that you wanted. And Rebecca would come out with all these stacks of books on weird subjects and horrible diseases that were like nightmare inducing. So I think that probably I contributed at least in that way. But we're definitely science girls. All of our girls are very, very science oriented. love it.
What kind of books did Melissa come out with?
You know, Melissa was very into Carl Sagan for a long time, and I think probably still is, but
Yes.
just anything nerdy. I remember one of the youngest daughter, you know, she is sitting wearing a t-shirt that her chemistry professor at high school had designed. Do you remember what it said, Rebecca?
No, we do have we have a little sister too. So I don't remember what you're talking about.
So there was some t-shirt and it was something ridiculous about, you know, math and being a nerd. And you know, I said, what are you doing? She said, well, Mom, I'm studying. Can you not read my t-shirt? I'm in band and I like math. So I think they're just all super interesting individuals with great imaginations, good creativity, which I think really lends itself into the science field. I think you really do need to be creative to be in science. And I'm super proud of them both, all of them. Yay, Becca.
Way to
Thanks,
go.
Mom.
You're welcome.
And your younger sister, what's she up to today?
She's in California studying math. So that's all I want to say about that.
And are you still living in Georgia?
No, I'm in Alabama now.
Oh, warmer Alabama, yes.
With 73 today, y'all ice people.
Yeah, thanks. We call that a 15-yard penalty for taunting, but that's okay. We were up in the 40s yesterday. So that's
I
pretty good.
understand that y'all break out the shorts when it hits about 40.
Well, I have shorts on right now, but I'm not going to back up and show you, but that's all right. You know, you don't want to get heat stroke on your legs when it's up there in the 42, 43.
Oh, Lord.
Yeah.
All right. Well, congratulations, everybody. What a great presentation. Good job, Rebecca. I was very, very impressed. Really interesting.
Thanks, Mom.
And thank you for all three of you for making it a family event. I really appreciate that. Okay, Dareth, you got anything? Dareth's a long time Wednesday night at the Adler.
My family has a saying that I think might apply to Karen's. Nerds, children of nerds. And we say it with pride.
I hear that.
All right. I'm gonna chime off here then and end it and end this Zoom, and thanks again. Hopefully we'll be able to have all this, the Q&A and the YouTube recording, and we'll see how it goes.